This is a partial chemical structure of DNA. It consists of thymine, adenine, cytosine, and guanine. They are the bases, and they're all connected by the hydrogen bonds labeled right here.
So this is a 5-carbon molecule, which is our deoxyribose sugar right over here. As you can see, the carbons are labeled with a pink marker. So here we got 1 carbon, 2, 3, 4, and 5. And right here is our phosphate group right over here. So to demonstrate how DNA is replicated semi-conservatively, we'll be using this model. So in the first step of DNA replication, separation of DNA is crucial. And this is the helicase, which is an enzyme that's in charge of splitting or separating the DNA strand. And thus it creates a shape called the structure called a replication fork. And each strand of DNA that is split provides a template for DNA replication. So the first steps, elongation, the primase starts the process, as you can see, the DNA primase. This is also another crucial enzyme in DNA replication. This enzyme creates the RNA primer, where the DNA polymerase can bind to, and therefore the DNA polymerase will start start reading the DNA from 5 to 3, and therefore that over here shown will replicate the DNA. That demonstrates the leading strand. However, the lagging strand is a little different story. As you can see, this is read backwards. So in our video later on, we'll explain why this is different. The lagging strand is different than the leading strand. And finally, the ligase, which is also another crucial enzyme, it seals up the fragments of DNA and therefore ending the process of DNA replication.
This is a demonstration of DNA replication with its parent strand and how it splits into the leading strand and lagging strand. So right here, this is the parent strand, the top is the leading strand, and the bottom is the lagging strand. So the leading strand, the template will be reading in 3' to 5' direction. DNA polymerase can only add sequential nucleotides in the direction of 5' to 3'. The leading strand is continuous. So the lagging strand is discontinuous and it is replicated in fragments shown right here. The lagging strand template will be reading from 5' to 3' direction, while the DNA polymerase will go in 3' to 5' direction. At the same time, the DNA helicase will unwind the double helix, which is right here. The RNA primase will add RNA primer, and the DNA polymerase will bind and synthesize small segments of DNA. So we have these fragments called Okazaki fragments, and they're small segments of DNA that are extended by the DNA polymerase, shown right here. So why does DNA shorten with each copy? In eukaryotes, we have linear DNA. DNA shortens with each copy because during cell division, the chromosomes are shortened by about 25 to 200 base pairs per replication, while the cell carries out DNA replication. DNA is undamaged because the only part of the chromosome is lost is the telomeres. So telomeres are located at the end of chromosomes. And human telomeres carries the sequence of TTA, GGG. Without telomeres, important DNA would be lost every time the cell divides, and it will eventually lose the whole entire gene. So why don't bacteria need to worry about this? So bacteria are prokaryotes. In prokaryotes, they have circular DNA. So during bacterial cell division, it's much more simpler and more efficient compared to linear DNA. circular DNA lacks telomeres, therefore it doesn't get shorter with each replication cycle.
So, how do we actually fit a very, very long strand of DNA into a very, very small chromosome? So, in order for us to package such long DNA, we have these histone protein molecules, which we need 8 in order for us to form a nucleosome. A nucleosome is basically when the DNA, as you can see in this picture, the DNA wraps around the histone protein, and since there is 8, it will form a cylindrical shape, and then as the DNA wrap around it, it will form a nucleosome. And nucleosomes are the basic small coils that forms the chromosomes, except the nucleosomes are also further condensed and also further wrapped in order for it to fit more DNA into the chromosome. So how does this relate to the expression of genetics? So modifications to the histone proteins actually inhibits or promote the coiling or the condensation of the chromatins, which can lead to changes in the chromatin organization, which affects how the associated genes are expressed.